Hello, class. Today we'll be discussing the topic of creating a new species, aka speciation. Early historical precedents. As do many conversations in the biological community, this one too starts with a famous scientist by the name of Charles Darwin. In 1859, Charles Darwin coined the term specification in his The Origin of Species. And it wasn't until 1906 when this term was recoined speciation by a biologist named Orator Cook. In this time, the middle of the 19th century, many naturalists recognized the relationship between the way species are distributed and their evolution. However, within Darwin's The Origin of Species, there were some major points of contention. Not only was it controversial for the time, um, as far as the populace is concerned, but within the scientific community, Darwin also came under fire from many renowned scientists of the day, two of which being Thomas Huxley and George Romanes. Huxley and Romanes not only had an issue with the way Darwin didn't address many of the biological questions, one of which being why hybrid species are sterile. Darwin also didn't address the title of his work and didn't explain specifically the origin of species and what that actually meant. Um, and then furthermore, there was also a biogeographic influence in this theory. Although other scientists came before him to show biological differences between similar species that were separated by large bodies of water, um, to where populations were there seemed to be a mainland species and an island species. It wasn't until uh, a naturalist by the name of Leopold von Buch came along and was the first to recognize that geographic isolation may lead to new species. And this was reconciled in 1825. These ideas developed painstakingly slow, and until the 1930s, the era of modern synthesis, um, and it wasn't until this time when the names such as Fisher, Haldane, Wright, and Dobzhansky came along to really change the perspective of speciation. Here, these scientists developed mathematical population models to illuminate the genetic mechanisms of evolutionary change. And this started with uh, a man by the name of Ronald Fisher, who described disruptive selection acting on sympatric and parapatric populations. And this was seen in his work, The Genetical Theory of Natural Selection in 1930. His work was so before its time, other scientists of the era, such as Haldane, J.B. Haldane, and Sol Wright, completely disregarded it and did not take it for what it really was. And it wasn't until Theodosius Dobzhansky came along to where opinions began to change. In 1937, he published his work, The Genetics and Origin of Species. Within this text, he became one of the primary, or from this published findings, he became one of the primary contributors to the incorporation of speciation into the modern synthesis. Dobzhansky was a geneticist, giving him a little bit more insight to this field. Here he formulated the genetic framework of how speciation could occur. His premise rejected Darwin's idea of speciation, which was that speciation was occurring through the occupation of new niches. Instead, Dobzhansky claimed that speciation that occurred due to an isolation of gene flow. After this, um, we see we see Ernst Mayr come and tie everything together. Ernst Mayr conducted extensive work on the geography of species and emphasizing the importance of geographic separation and isolation, and so filling in the gaps that Dobzhansky left, especially those concerning the origin of biodiversity. And here we see that in his in, in two of his major works, Systematics in the Origin of Species from the Viewpoint of Zoologist and Animal Species and Evolution, published in 1942 and 1963 respectively. And these two, Dobzhansky and Mayr, without controversy, gave rise to the understanding of speciation, stimulating research and growth in the field for decades to come. Types of speciation. There are four main types of speciation. The first being allopatric. Allo means other. Patric means place, so other place. This type of speciation occurs due to geographic isolation. As we sort of touched on earlier, speciation can occur when these two gene pools become separated and they start passing on their genes respectively. And these two groups become separated by a physical barrier of some sort. It can be something as small as a tree for a species very small, or population is extremely small, or for larger species, we typically see bodies of water or mountain ranges. Furthermore, allopatric speciation is not to say that certain individuals can navigate these barriers to make it back to the other population, but is to say the majority must stay separated from each other. An example of this would be Darwin's finches. The next form of speciation is peripatric. Peri means near. This is a special type of allopatric speciation or isolation in which some instance has occurred, causing the majority of the population to die off, leaving a very small population with a limited gene pool. And due to this limited gene pool, rare genes are allowed to be passed down, causing genetic drift. And this is illustrated all on the image on the right. The next form of speciation is parapatric, para meaning beside, and then patric meaning place. Within this form of speciation, populations are not isolated by any physical barrier, but for some particular reason, individuals only breed with those in their immediate area. An example of this would be orioles, as there is a hybrid species that exists in Texas and in the U.S. state of Texas and Oklahoma area, and then we see two completely different species to the left and right, or east and west. To the east being the Baltimore oriole, and to the west being the Bullock's oriole. The fourth type of speciation is called sympatric speciation. Sym means same, and patric meaning place. Here, this form of speciation is here these individuals are not separated by any geographic or physical barrier. Populations are instead isolated through reproductive barriers or reasons. Many species, there's a few ways this can occur. There are many species that return to where they were born in order to breed, or populations breed in different seasons. So as you can imagine, as certain populations return to where they're born, this sort of this constricts the gene pool to only those that are near it. It's sort of how we see in the last form, but the reason is all reproductive and biological in that sense. And this brings us to our last term of speciation, and that being artificial speciation. This type of speciation is probably the most familiar to us because it is the one that humanity directly plays a role in. For thousands of years, humans have had their hand in domesticating and interbreeding animals in specific ways in order to achieve desired traits of those species. We see this in horses, dogs, cattle, and many others that we've domesticated. Through controlling which individuals breed with which, we are able to have a direct impact on the genome of that domesticated species. Today, things have taken quite the turn, and we have seen a different model, a modern application in the form of genome editing. And so, as you can see on the slide, Kelsey J.R.P. Byers and H.D. Bradshaw Jr. Um, they conducted experiments on hawk moths through altering a flower's pollinating syndrome. A pollinating syndrome is essentially the flower's means of being pollinated. This includes the petals, um, the shape, the length, every mechanism that goes into pollinating a flower. And here, these scientists found here in their published work of Frontiers in Ecology and Evolution, um, they found that through changing a flower's phenotype for color, the flower being monkey flowers, from red to that of a lighter color, pink, white, and in the colors in between of those, they were able to attract an entirely new species to this flower. That being a hawk moth, which, unlike a hummingbird, cannot recognize deep shades of red as that being a potential food source. So, what this means is that if we have the ability to to edit or alter the pollinating syndrome of certain plants or flora, we can have a direct impact on how the organisms that interact with these flora um, change and evolve and uh, and adapt to their new or these new food sources. And as they adapt to a new potential food sources or that or lack of those, changes will be made within the species in order to better accommodate for that. And in a sense, and the possibility for creating an entirely new species has been discovered with only playing an indirect impact. And uh, this study was conducted as of 2021. So findings on this are going to continue to develop and continue to change the way that we look at the way we impact our environment and the way we can affect it. Lastly, here are my citations, and from all the work that I did. Uh, Thank you. And this has been my brief intro into the topic of creating a new species, aka speciation. Thank you.